children. Today in this video we are going to start our new chapter, that is seed structure and germination. So what is a seed? So before we talk about the germination of seeds, it will be very important to know the terms such as fruits, seeds and grain. Okay. So what is a fruit? Fruit, it is enlarged ripened ovary. The ovarian wall forming the fruit wall which encloses the seed. The fruit wall or the fruit, what it does, it protects the seed and helps in the dispersal of seed. For example, mango and pea pod. Now the next term which we are going to study is seed. Seed is the ripened ovule. Fruit is the ripened ovary and seed is the ripened ovule. This seed, it contains an embryo and that embryo develops into a new plant. And the outer covering of the seed, which is known as seed coat, what it does, it protects the embryo from any other mechanical damage. For example, we can see in case of bean seed, peas. Okay. Now a third term is grain. Where these grains are found? These grains, they are found in maize, wheats. So these grains, what are they? They are actually the fruit in which the fruit wall and the seed coat, they both are mixed together or fused together and they form a protective layer. Now let us study more about seeds. What is the seed? We have discussed now. The seed is the ripened ovule. When it is the ripened ovule, it is a mature ovule after the process of fertilization. What is fertilization? We have discussed in the earlier video. Fertilization, it is the fusion of the nuclei of male and female gamete. So this seed, it is mature ovule after fertilization. And this seed, it contains a very small living plant which we say as embryo. Okay. This embryo, it remains in an inactive state. That means this embryo, it remains in a resting state until when this embryo will be getting favorable conditions. So when this embryo will get favorable condition, it germinates. Now this seed, it also contains a food material for the nourishment of the embryo during the process of germination. And this embryo, this can withstand unfavorable conditions of temperature, drought, etc. Okay, some of the seeds, they are known to remain dormant for so many years, that means hundreds of years or more even. Okay, now come to the next part, that is classification and structure of seeds. On that basis, there are two types of seeds. First is monocotyledonous seed and second is dicotyledonous seeds. Okay. Monocotyledonous, mono means single. That means seeds in which only one cotyledon is present. Suppose we can take the case of maize, grass. Dicotyledonous seeds, that means in which two cotyledons are present. For example, pea, bean. Okay. Now seeds, they are of many sizes. Some are small, some are large and some are largest, such as we can see in case of coconut. Some are small, poppy seeds. Okay, some are quite large, that means in watermelon. So the size, shape and structure of the seeds of the different plants may vary, but the basic structure of most of the seeds is same. Now on the basis of endosperm, again these seeds, they are classified into albuminous and ex-albuminous. albuminous, that means in which endosperm is present. X-albuminous, that means in which endosperm is not present. So albuminous seed, it is known as endospermic seeds, in which the cotyledons are thin. Cotyledons, that means these two structures. These are the cotyledons. So in albuminous seeds, the cotyledons are thin and membranous, that means they are having layers. And endosperm is also present, that's why they are known as endospermic. They again are two types, that means dicot albuminous and monocot albuminous. Now X-albuminous, that means non-endospermic seeds. So in these seeds, the cotyledons, what they do, they store food and they become thick and fleshy. So when they store food, these cotyledons, they become thick and fleshy. Again, they are of two types, dicot X-albuminous and monocot X-albuminous. Now come to the structure of bean seed. There are a number of different kinds of bean seeds, such as broad bean, lima bean, French bean, etc. Okay, but the general structure of the seeds, as we have already discussed, the same. So most of the seeds, they are kidney shaped. Okay. Now this seed coat, it consists of outer layer, which is known as testa. This is the outer layer. Okay, so this seed coat, it consists of testa, which is the outermost hard brownish covering. And what is the function of this testa? It protects the inner part, as because it is the outermost layer, so it is protecting the inner delicate parts of the seed from any type of bacterial attack, from any type of injury, etc. Okay, now the next layer is, second layer is tegmen. It is thin inner layer next to the testa, and this is also a protective layer. It is also covering the inner parts. Now hilum. Hilum, it is a white oval scar on the concave side of the seed. Here this scar is present. Okay, and what is the function? It is the spot where the ovule, which is now the seed, okay, it is attached to the ovary wall. This ovule, which is now has become seed, it was attached to the ovary wall with the help of placenta. Now a small pore, which is known as micropyle, it is also present, and closed, it is present close to this hilum. Okay, so the function of this micropyle is it absorbs water when the seeds, they are soaked in water. The seeds, what they do, they absorb water. So from where these seeds, they absorb water? Through this micropyle, and make it available for the embryo, the process of germination. Okay. The second point is diffusion of gases. Diffusion of gases, it provides for the diffusion of respiratory gases for the growing embryo. So this micropyle, it marks the opening through the pollen tube, through which pollen tube it entered the ovule. Okay. Now what the function of micropyle? Two functions: absorption of water and diffusion of gases. Now below the seed coat, we have seen there are two cotyledons present, two thick cotyledons present. And what is the function of cotyledon? It's provide, or it contains food for the embryo and protect the embryo also. What happens when we separate these cotyledons? Okay, so the tiny embryo can be easily, see, this is separated cotyledon. When we separated these cotyledon, a tiny embryo is seen inside it. Okay, attached to this tiny embryo, it is attached to the cotyledon. And this embryo consists of two parts: the upper part, that is plumule, and the lower part, which is radicle. Okay. Now this plumule later forms the shoot system, and radicle, it later forms the root system. plumule, it consists of a very short, this plumule, it consists of a short stem, and it is having pair of tiny leaves, 1, 2. See here, these 2 leaves are present at the growing point in between, and through that this plant will be growing. So this was all about the structure of a bean seed. Dear students, so that was all about the structure of the bean seed. So in structure of the bean seed, what we have studied: seed coat, it is having 2 layers, testa and tegmen. Hilum is present, whose function is to attach. It attaches ovule to the ovary wall. Micropyle is present. It has 2 function: absorption of water and diffusion of gases. Cotyledons, which is having food for the embryo and protecting. So this was all about structure. Then in the next video, we will study the structure of maize grain. Till then, student, revise this and take care.